So good afternoon, guys. It's Saturday afternoon. Sorry, I've been running crazy. I'm going to walk through chapter 7 kind of quickly, and then maybe get to 8 tonight, today too. All right, so hope y'all getting your online homework done. Let me know if you're having problems. Here we go. I'm going to flip the screen over to here. Okay, you should see. Yeah, should see it here. Everybody see that? Okay, should see chapter 7. Okay, here's the breakdown of the different parts of the chapter. We're going to be looking at photosynthetic organisms, process photosynthesis, how the plants convert solar energy into usable ATPs. We'll see fixation of carbon dioxide, and then a couple of the other types of photosynthesis that we'll talk about. They're kind of different. Okay. All right. So first of all, we talked about different plant science, tweaking the basic chemistry here. Nothing much on that one. I'm just going to get on into the whole point of it. Is that all life on Earth depends on solar energy, right? We talked about this in class. Either we eat a cow or a pig that ate the plant, and the plant is able to do photosynthesis and capture this solar energy, and thereby provide energy for everybody else. If you remember, organisms that eat somebody else are heterotrophs. Organisms that make their own energy are autotrophs, and so the plants are photosynthetic autotrophs. Okay. Couple of cool terms you should probably know at some point. Okay, now we're going to see that photosynthesis takes place obviously in the chloroplast and the leaves of the plant. Here's our nice circle of life here, so we can see that, remember, carbon dioxide and water will be taken up by the plant, and they will use this to use sunlight through the chloroplast organelle to produce glucose and release oxygen, right? And then animals and other organisms need that sugar and oxygen to do cellular respiration with the mitochondria. Now, our next chapter is going to be talking about the mitochondria, but interesting to note here that in photosynthesis, super important here, there is no net ATP made. They make sugar, and then the plant will turn around and use that sugar in their mitochondria to break it down to make ATP. So I use the cliche a lot in class that the plants make the cake, the sugar, and then they eat it too. They have a mitochondria, and through cellular respiration, chapter 8, they're able to get energy from it, as we'll see. Okay. All right, so let's dive into the leaf. Here's a cross section of the leaf. You can see that these they have epithelium tissue, right? They have the hole we're going to talk about in the bottom of the leaf where they do their gas exchange called the stomata, and carbon dioxide is coming in here and oxygen is leaving. There's a picture kind of showing what it would look like from the side view. And inside these little green discs called the chloroplast, we will see that these are inside there. You can see the little green thing. These are the organelle where photosynthesis occurs. It has two main components. These are the green stacks of thylakoid membranes called granum. Each disc is called a thylakoid membrane. And then the cytoplasmic area or fluid part called the stroma. Okay. And then we'll see that, actually, sorry, I didn't know that I hit the wrong thing there. And you can also see there's the thylakoid space inside the thylakoid membranes, right? And they overlap and they have openings that allow. This is an electron microscope at 23,000 power. And then we've got a drawing there on the side. Okay, now, basically, very simply put, that photosynthesis is going to be two major metabolic pathway reactions. The first one is called the light reaction, and it is going to occur on the granum, on that thylakoid membrane, and it's going to use the sunlight to create ATP and another compound called NADPH, which if you remember, NAD and NADP are energy and electron transfer molecules in photosynthesis and cellular respiration. NADP is in the mitochondria. NADPH is the electron transfer molecule that we see in the plants. Okay, so we're going to have this light reaction occurring, right? And we're going to see, you can kind of read along here that we're going to also have electron transport chain in here. And I'm going to show you the details of that. And the bottom line is we're going to make 18 ATPs and 12 NADPHs in this light reaction. Then and water is used to provide the electrons that are going to be found in those NADPHs, okay, which represent high energy electrons used to build the sugar. And then the Calvin cycle occurs in the stroma, and it is going to be mainly the reduction of carbon dioxide to build a sugar to make the carbohydrates, right? And we're going to use up all 18 ATPs and all 12 NADPHs made in the light reaction. Sometimes this is called the light independent reaction, meaning the dark reaction. You can play a metaphor on light dark here. And also, Calvin was the PhD professor who, graduate student Benson, I think, did his PhD on this, discovery of this, a Nobel Prize, I believe. So sometimes it's called the Kelvin-Benson cycle. Okay. All right, and you can see here is some of the products of some Eladia. There's the bubbles of oxygen that they release as they do photosynthesis, and that's pretty cool. Okay. So once again, let's look at it again. Solar energy is going to be captured in the granum, and it's going to be used to make ATP and NADPHs, which are going to be high energy electrons. There they are. Okay. NADP is carrying high energy electrons, 12 of those, and 18 ATPs. And then this light independent Kelvin-Benson cycle, the carbon dioxide is going to be reduced to sugar, and we're going to use all this energy up. And as we know, plants use that sugar for energy with their mitochondria, or they can use it to build their structure, their body itself. Okay. Interesting enough, the electrons that are in the NADPH that end up in the sugar come from the breakdown of water, and the oxygen released to end the light reaction is from the water. Okay. All right, we're going to look at this now and go into a little bit more detail. Now, your next part of the chapter, it talks about these photopigments and the photosystems, okay? And I'm going to try my best to explain it. I think I did a pretty good job in class. So a lot of this is going to be reviewed, but hey, can never hear something. It's never hurt to hear the same thing twice. But the thing I think is super cool here is that these photopigments, mainly you've heard of them, chlorophyll A, chlorophyll B, the carotenoids and xanthias. These photopigments are very special chemicals that are found found in the plants, and are found in some bacteria, and they have the ability to take the energy from the sun called photons and absorb that energy and take an electron and excite it. And when I say excite it, I mean take it to a higher energy level, like from the first shell out to the third or fourth shell. And in doing so, that energy is transferred from solar energy into potential energy in that electron in an outside shell. Okay. So we're going to go look at these photosystems in just a second, which is a packet containing all these photopigments inside the membrane of the thylakoid membrane of the granum and the chloroplast. Okay, but before we do that, we kind of look at the absorption spectrum. This is kind of cool. And if you remember recalling us talking about this in class, that big red ball in the sky called the sun emits energy in packets called photons. These photons contain gamma rays, X-rays, ultraviolet rays, visible light, infrared, microwaves, and radio waves. Okay. And what we can see here, and y'all have heard about gamma rays, right? Those are really dangerous, and our atmosphere helps absorb that, filter that out. X-rays can be dangerous, right? But we use them medically, right? To look inside and see if bones are broken. And then ultraviolet rays are the ones that cause us to tan, right? And then we talk about that. That's kind of cool. But that visible spectrum is 138 to 750 nanometers. Notice now that the waves get bigger as we go from left to right. The smaller the wave, the more deadly, and the more increase in energy it has. So these smaller waves are much more deadly than these radio waves and TV waves that we're being microwaves out here in nature that we're being bombarded with all the time. Your cell phone uses microwave waves that we think are safe, okay? And don't have it, but a lot of people are scared or worried about that, but that's another story. Look at this. This is kind of cool. This is the where you see the peaks. This is the wavelength of visible light that plant likes. Now, I think one of the quick fun side notes about visible light is that the white light coming from your bulb is actually a mixture of all these. And if you take a prism, not prism where Uncle Billy's at, but prism, the little piece of glass that you shoot light through, it will break it into the rainbow. Okay, and so the rainbow actually goes from purple to blue to green to yellow to orange to red. So the wavelengths are different colors of the light. A visible light has different colors in it. And certain plants and certain photopigments like certain wavelengths. For example, chlorophyll A loves like 400 purples, right? And it loves the red and orange, okay? Chlorophyll B likes a little bit more on the yellow-orange side and the blue to green. I mean, blue to blue, I say blue here. And then carotenoids can go a little bit almost into the green, but notice that green is the main color the plants don't use. And this is because what you see is what is reflected. So my shirt is red today. It's reflecting the light from the bulbs in here going to the camera, and you're seeing a reflection of the red. It absorbs all the other colors, okay? And so black absorbs all colors, and it's hot on a sunny day, okay? But white reflects all colors, and it is cooler on a sunny day, a white shirt, okay? And so I think it's kind of ironic that plants do not use green, which is the color they are, which we associate with nature and green peas and kumbaya and all that good stuff, all that kind of tree hugging stuff, right? But green is not the color they use. They use the orange, reds, blues, and purples, okay, to excite electrons, thereby to do photosynthesis, okay? And I think that's good stuff, okay? Now, How do we do this? Well, we said that there is the two steps to this, right? There is the light reaction we're going to talk about now, and the light independent Calvin cycle. Well, come to find out, there are two types of light reactions. Okay, one is called non-cyclic, and that's the most common one that we see out in nature in all the plants in the backyard, and we mow the grass and butter beans and corn. And then the cyclic pathway is one that we see in algae, and it was actually one of the first ones discovered. Okay, and so it's and we'll see that inside the chloroplast there are two photosystems, okay, that are going to be absorbing the solar energy. Remember, the photosystem was a packet of chlorophyll and these photopigments that has an antenna that is set up to catch the energy from the sun. Well, interesting enough, the one that does the cyclic pathway out in nature was discovered first, and it's named first, and the first one in the sequence was discovered second, and it is photosystem 2. Let me show you a better picture. I'm probably being very confusing here. I'm just going to go on to over here. We done looked at this. This is our summary. But let's go to this picture right here. I think this does a little bit better, and I actually like seeing it in the granum itself in just a second. But here's what's going to happen. These photopigments. So let's just go ahead and look at it. This is so crazy. I just think seeing the whole thing better and doing it backwards. I don't necessarily agree with the order of this in the chapter. Here is this photosystem, and that contains that antenna. A photosystem is a pack containing all these chlorophyll molecules and these photopigments arranged in an antenna to catch that sun energy and excite an electron in what's called the reaction center electron. Okay. As a matter of fact, you can see it right here. I'm going to back up just a little bit. You can see it right there. There's the reaction center. Here's the antenna array, and that solar energy is hitting it. And in doing that, water is broken down into oxygen and hydrogens, and the electrons are pulled off, and then that chlorophyll molecule in the middle gets all the energy from all these other chlorophylls that the sunlight's hitting them, very much like a direct TV antenna works on your house, and that electron's excited to a higher energy level. Then that electron is going to go through what's called an electron transport chain, okay? And this is a series of proteins stuck in that granum, the cell membrane of the granum, the thylakoid membrane. And in doing that, that energy is transferred from each molecule to another. And in doing that, some of the energy is used to pump hydrogens into this inner space. Now, let me show you that right here. So here it is. Here is photosystem 1. Electron gets excited. Then it goes through this electron transport chain that pump hydrogens into this thylakoid space. So we end up taking them from an area of low to high, concentrate them, and they want to go from high to low. And we're going to see that on that membrane is a pump, protein pump, called ATP synthase. It actually has a propeller in here that turns, so as the hydrogen go through, they turn the propeller and they staple a phosphate onto ADP, thereby making ATP. Okay, pretty cool. And when you break down 6 waters, you make 18 ATPs this way. Okay, so bam, a lot of hydrogens get pumped. When that electron gets worn out, it goes to photosystem 1, another antenna, juices it up, gets it hot again, gets it to a higher energy level, and then an enzyme called NADP reductase then gives it up to NADP, and now we have NADPH, which is the reduced form. Remember, oxidized means to lose an electron, reduced means to gain. So NADP is reduced to NADPH, and you end up making 12 of these for 6 waters being carried through. And that represents high energy electrons that are needed to build the sugar in the Calvin-Benson cycle, also called the light independent reaction. Okay, I kind of like seeing it, and it's real. This is it. This is where it's really happening. That's what it looks like. Okay, I'll back up. I'm having issues with my backup here. So the electron then runs down the ETS. Photosystem 1 grabs it, excites it again, and that electron ends up as NADPH that will then be used in the Calvin cycle. All of them and all the ATPs made here will be used up in the Calvin cycle, and NADP will come back and grab more electrons, and ADP will come back and get recycled to ATP. This is non-cyclic because electron starts in water and ends up in the sugar, so it doesn't circle. In the cyclic reaction, the other type of photosystem, you only have photosystem 1, and this electron gets handed right back to the antenna. So it just makes a circle, and in doing that, you end up pumping hydrogens and making ATP. So basically, you have this without NADP reductase. You just have another electron transport chain here, and you make energy. And this is seen in algae that don't have big bodies, that don't need lots of sugar, but they need ATP. Okay, so there it talks about the photosystem. 2, that's the first one that releases the oxygen, breaking it from water. And then photosystem 1 juices it back, electron back up to higher energy level, and it reduces NADP to NADPH. That's then carried to the Calvin-Benson cycle. Use all that ATP, 18 ATPs, 12 NADPHs are used to make the sugar molecule. And the ATP synthase is where, remember, it's where all the ATPs are made. Okay, there it is, bingo. And now we're going to, I think we're ready to talk about we're going to slide into the Calvin-Benson cycle in just a second. This is just showing you how the electrons go through ATP synthase, the hydrogens, and that's used to make the ATP. And this is called chemoosmotic ATP production. Now, you ready for a mind blower? Next week, or this coming week for y'all in lab, because I'm going to probably make this video today, the other one for 8. Guess what we see in the mitochondria? Same thing, electron transport chain, a ATP synthase, and we're going to see in the mitochondria that we're going to make 32 ATPs there using a very similar process. Okay, so it's got, and so because of that, we believe the chloroplast that we now think was a separate microorganism that came to live inside the bigger cell, and the mitochondria were probably kin before they each became the chloroplast and the mitochondrial organelle. Okay, which is pretty cool. Okay, and then there's good commentary in here, and I think I have a question on the short answer list about this, but tropical rain, forest destruction, and its effect on global warming. Very good. Read that one. I got a short answer question about that on the review sheets, but fascinating how we're seeing this increase in carbon dioxide, right, over time, and how this is linked to global warming, okay? And so as we burn these forests down, that is the sink, or somebody, I think the book even said the drain where carbon dioxide is being pulled back into biomass. And as we cut these rainforests, the CO2 levels are not getting sequestered in wood and now are even going higher. So we've got real issues there. And we'll talk about, I think there's actually an ecology chapter later in majors too. Here's some of those numbers showing some differences on that. Okay, now we're going to look at the Calvin-Benson cycle. Okay, where we're going to do carbon dioxide fixation. That's where we bring it into the plant, right? And then we're going to have carbon dioxide reduction, and then ribulose biphosphate regeneration. Okay. Now that first step, carbon dioxide comes in and binds is attached to 5-carbon sugar called ribulose biphosphate. An enzyme, ribulose biphosphate carboxylase, is in a lot of books considered the most common compound on Earth. Many chloroplasts, half their weight is this enzyme, and our book says that it's very slow. So it's this enzyme called reaction accelerated by that's called Ribisco is thought to be a very slow enzyme. So it only does a few molecules an hour, whereas most enzymes do hundreds per second. Or maybe a few molecules per second instead of 1,000 per second. It is thought this may be the most common compound on Earth, or most common organic compound on Earth. Okay, but that CO2 is fixated, and I think one of the cool things when you look at this Calvin-Benson cycle is you can follow the carbons. Remember, the first law of thermodynamics says we cannot create or destroy anything, we can only rearrange it. And so what we're doing is you can count the carbons. We're not making any carbons, we're not destroying carbons, we're just recycling them around, and we're building this sugar molecule, okay? Okay, so here we go, Calvin-Benson cycle over here to the sit there on the right. Okay, first step is 3 carbon dioxides comes in, and that's 3 carbons and 3 ribulose biphosphates, that's a 3-carbon sugar. So that's 3 times 3 is 9, plus 3 makes 12 electrons, right? 12 carbons, excuse me. And you get some intermediates, but you end up with 6 3-phosphoglycerates, which is 3 times 6 is 18. We're going to use 6 of those 18 ATPs, and you end up with 6 biphosphate glycerates, okay? And then we use 6 NADPHs, okay? And you end up with 6 glucose 3-phosphates, okay? Okay, or actually, is that right? Yeah, glyceraldehyde 3-phosphate, excuse me. They got it spelled right there for me. I didn't see it. So there's the actual, tell you the compound as the intermediates. You're not got to know any of those, but I do want you to know what goes in, what comes out. Remember our rule? We talked about that, okay? Now, look what happens. This thing has to run, and your book keeps saying it has to run 3 times to get those 3 through. So after it does that, look what happens. After it runs 3 times, or it goes to right here, it does 3 carbon dioxide, so that's 3 times. Then you have 1 glycerol. You end up with these 6 glyceraldehyde 3-phosphates. 1 of them is stored back, and then it's got to happen 3 more times, and you get 2, and 2 of them put together to make a glucose. Okay, so it takes 2 of those. So really, we'll see in just a minute, we got to double all the ATP and NADPH amounts shown here. Then look what happens. The other 5, now 3 times 5 is 15, right? 3 times 5 is 15. The other 5 then are going to slide over here and use 3 more ATPs and regenerate 3 ribulose biphosphates. Okay, which if you'll look at that, remember, look, those are what we said. Those were 5 carbon sugars, right? So 3 times 5 is 15, and so look at there. There we go. 3 of those times 5 is 15. So we don't create, but you have that one glyceraldehyde 3-phosphate that's been pulled back, right? That's very important, right? Because it has those 6 carbons, right? Okay, or 3 carbons, and then we're going to stick to them together to make glucose. So for the whole thing to happen, and then it reboots itself, so it's a self-generating circular metabolic pathway, and we'll see one very interestingly, a circular metabolic pathway in the mitochondria to break sugar down. But here in the stroma of the chloroplast is this circular metabolic pathway to build sugar. So this whole thing actually got to run 6 times because there's 6 carbon dioxides. This is just 3. We got to have 3 more come through. So you end up using 8, 9, 18, all 18 ATPs, and then what we got here is it 8 ATP? I may have my NADPH number wrong. It actually that number, that is 6. I can't see it here. So 6 are used, and I said there were 12. So that the other 6 are used. So the whole thing, it runs 3 times, and you make 1. What is it? 1 G3P, which is our glyceraldehyde 3-phosphate. 2 of them stick together, make a glucose. And then the other thing that's cool is plants can then use this to make all kind of sugars, fats, amino acids, and starch, cellulose for structural part, branches, roots and stuff, and then sucrose in the leaves, fruits, and seeds. So they make all their sugars using that. And then the other thing to remember is they make so much sugar is that there's no ATP left. Then they burn the sugar, the glucose, to make their ATP in the mitochondria, which we'll talk about in chapter 8, the next chapter. So this is, to me, it's kind of cool. Okay. And so they kind of walk back through that. And that's my best way I did my best to explain it. But they talk about the fixation again, the reduction of the carbon dioxide, right? The NADH being used by sticking those electrons off the NADP and putting them on the 3 PGs. You make these, you make these glyceraldehyde 3-phosphates, right? Which have more stored energy in them. Okay. And it talks about the rebooting it back to the ribulose biphosphates to make 5 of those to grab another carbon dioxide. And they're showing that. So that takes 3 ATPs, so we burn all 18 ATPs. Okay? And plants can make all those other things from that. Okay? Now, if that wasn't enough to make you scared, that was a tough one for me. Well, come to find out, there's other types of photosynthesis. And I'm actually looking over it, reading over it, because it ain't my super duper area of expertise. So I'm going to try to read a little bit out of the book to you here and show you this too. The majority of plants carry out what we call 3C photosynthesis, which is what I just showed you. Okay? And these use ribulose biphosphates, carboxylase, to fixate carbon dioxide to ribulose biphosphate in the mesophyll cells. Now, that is the cells of the leaf. Okay? These are the cells of the leaf. So these C3 plants and all those chloroplasts there. Now, remember, this is the vein, the leaf vein, which contains xylem and phloem. Phloem carries the sugar down the plant, and then the other one is the phloem, xylem. Excuse me. Phloem carries sugar down the plant. Xylem carries water and gases sometimes up and down the plant. So this is open to the stomata. Okay, so the stomata is where the gas exchange is going to occur, right there. See it? So this is the leaf veins, and there's that stomata. Now check this out. This is really cool. Okay, so C3 plants do what we just talked about. Okay, they do that. But here's the problem. The stomata must close to avoid wilting when it gets really hot. Mississippi, July, August, September. CO2 then decreases, right, because it can't bring it in. Oxygen increases because it can't open the stomata to let it go. And this oxygen starts combining with the ribulose biphosphate, leading to the production of carbon dioxide, and this is called photorespiration. C4 plants solve this problem by what I'm going to show you. Now, here's the punchline. That is bad for a plant, and that slows down the rate of photosynthesis, and this is why our grass yards, a lot of times our grasses in our yards start to slow down in July and August because they can't, this photorespiration slows down their rate to making sugar, and so the plants don't grow as fast. So to avoid the carbon dioxide being present around the enzymes so they can be converted so that the CO2, the low CO2, excuse me, so to do something with an oxygen and keep the CO2 apart, right? Here's what they do. The C4 plants, they're going to take carbon dioxide in and fixate it using a different enzyme to a 4-carbon sugar called oxalyl acetate, okay? So they're going to, and I'm reading it here, right here, they're going to take this C4 fixate carbon dioxide to phosphoenol pyruvate, a C3 molecule, and another enzyme converts it into oxalyl acetate. So in a C4 plant, CO2 is taken up in the mesophyll cells, and then malate, a reduced form of oxalyl acetate, is pumped into the bundle sheath cells, and only here does carbon dioxide enter the Calvin cycle. Now look at structurally the difference here. So this is occurring in this bundle sheath, and therefore the CO2 is not going to be photorespirated, right? And thereby, so in other words, what that's stopping is the photorespiration, which was when a molecule of the 3-phosphoglycerate eventually released photorespiration takes place when the carbon dioxide, let me read it again, oxygen rises in the oxygen combines it. So when oxygen combines with ribulose biphosphate carboxylase, it combines with ribulose biphosphate instead of CO2, and that eventually releases, this results in one molecule of 3-PG and the eventual release of CO2, and that is a problem, okay? So the oxygen is taken up and CO2 is released, okay? That's normal. So we want to stop photorespiration. That bundle sheath thereby is the only place, I'm reading it to you here out of the book, one more time, only here does CO2 enter the Calvin cycle. So in other words, the Calvin cycle in here occurs, so the CO2 is fixated, thereby preventing photorespiration, okay? Now, listen to the book, this is pretty cool. Because it takes energy pump molecules, you would think that C4 pathway would have a disadvantage. Yet in hot, dry climates, the net photosynthetic rate of C4 plants, such as sugarcane, corn, and Bermuda, is about 2 to 3 times that of C3 plants like wheat, rice, and oats. So why do C4 plants enjoy an advantage? The answer is that they can avoid photorespiration discussed previously. Photorespiration is wasteful because it is not part of the Calvin cycle, and photorespiration does not occur in C4 plants because this peptidase enzyme, unlike ribulose carboxylase, does not combine with oxygen even when the stomata is closed. So the CO2 is delivered to the Calvin cycle in the bundle sheath. So there is no oxygen being converted into the buildup of oxygen is not a problem here because the enzyme won't bind to it. So we don't get photorespiration in C4 plants. Very complicated. This is in your book. Hey, I'm reading it here and I think I probably screwed it up. But read over 125, 126 if you want to get a clear explanation for that. So it is a different pathway. So the CO2 is stored in as oxalyl acetate, and that's why it's called C4 plants, because carbon dioxide is fixated into the C4 oxalyl acetate. Okay? Kind of interesting, but this is why, come on, man, you live in the South. What grows good here when it gets hot? Corn and Bermuda. So your Bermuda grass does great when it gets hot, but if you got just regular grass like I do, it slows down in August, okay? Pretty interesting. And then if that ain't enough to make you want to just quit, there's another one, okay? And here it shows again, and that's the little enzyme showing that, how it works. So this carbon dioxide supply here is stored as oxalyl acetate in these cells, and that prevents the buildup of oxygen, causing problems over here and screws up the enzyme over here. And then we have another one, what I call CAM photosynthesis, and these are plants that live in the heat. I'm talking about cacti, and so CAM plants open up their stomata in the night, and carbon dioxide is fixed into a 4-carbon molecule and stored in large vacuoles, and so that they can then during the daytime, NADP and ADPs are available. Stomatas are closed for water conservation, and the C4 molecule releases CO2 to Calvin cycle, and then it can let oxygen out at night. So it only opens up at night to let the oxygen out and CO2 in, and these are our cactus plants. Okay, and they get that name. I think it's from there's a family of succulent desert water containing plants, jade plants that live in warm, dry regions where this was discovered. And they belong to this Crassinian acid metabolism, and the name of the family of the plants is Crassinian. So that's the C and the CAM. Okay, so that's another way. And that was easier to me to understand because they only at night open up, take the CO2 in, and fixate it into a 4-carbon sugar so that they can do Calvin cycle in the day when all that ATP and ADPH is available. Woo, pretty wild. And it talks about that. Cam 4 plants work better, have it adapt and are more adapted to high light intensity, high temperature, and limited rainfall. C3 plants do better in the cold below 25 degrees centigrade with high moisture. Cam plants are adapted to extreme dryness. Cams occur in 25 families of flowering plants. They're also found among non-flowering plants, or like our cactus. That's what I think of. All right, guys. I hope that was not too confusing. Sorry about C3, C4. That always gives me trouble. That's not my super expertise, but I do find this fascinating, photosynthesis. So y'all have a good one. I'll try to get chapter 8 up here in just a little while. Talk to y'all later. I'm going to stop this video, and we're going to call it a day on that one.